Thank you very much for the introduction, and thanks to the organizers for having me here, including the forum as well as the Academy of Sciences, and special thanks to Shyam Mani for including me in this elite group. It's always a tough act to follow people who work on the physics of stars, and it was a truly star-studded talk. But all I can say as a way of transition is that I'm reminded of Carl Sagan's famous quote in his very popular TV series called Cosmos, where he said, "Humans are made of star stuff." So with that little segue, I'm going to talk about, in a very different subject. And the irony is that much of neuroscience starts with the study of humans as a whole. And in that sense, human patients who have neurological disorders or deficits are often the stars of neuroscience. And I'll start off with giving you one example of a patient who the story goes back to more than 100 years. And this was a patient who was being seen by a Swiss psychiatrist, Edward Claparede, at the turn of the century. And he had this female patient who was completely incapable of forming any new memories. And although her older memories were preserved, but the problem was severe enough that she was unable to remember anything beyond the duration of a few minutes. And this was so severe that even though Dr. Claparede was seeing this patient many times a day for many months, she couldn't remember him at all. So this led to this curious situation where Dr. Claparede had to shake her hands every time they met, even if it was multiple times a day. And of course, Dr. Claparede got pretty tired of this and decided to try something different. So he hid a pin in his palm, and the next time they shook hands, he pricked her palm with the pin. This is something I don't think you can do these days, but that was about all they had in those days. So this was with the hope that maybe this bad experience would somehow jog her memory of him. Unfortunately, Dr. Claparede's experiment didn't work, because even after all of this, the next day when they met, she still didn't remember him at all or anything about yesterday. So there was a clear and very severe deficit of what I call factual memories, because anything related to that event was completely lost to her. Yet something very curious happened. Despite this very severe impairment of factual memory, she refused to shake Dr. Claparede's hand without being able to explain why. So she had this sort of no explicit conscious memory of the meeting Dr. Claparede, but she had this implicit and very strong subconscious fear that somehow shaking his hands is not a good idea, although she didn't, although she had no recollection of him as a person. So here you have a very curious situation where you have severely impaired factual memory coexisting with a very strong fear memory, all related to the same event. So this asymmetry is what I find quite fascinating. And this is something that we experience in our own lives. We don't have to go to a patient to see this. We struggle to remember phone numbers, emails, names, faces all the time. So factual memories are prone to decay over time, whereas an emotional event in your life, like happy or sad or tragic, will remain with you for the lifetime, even if it's only once in a lifetime experience. So in those days, of course, Dr. Claparede and his colleagues had no access to trying to understand what are the neural mechanisms subserving this kind of asymmetry. We now know, after the benefit of almost 100 years of research, that there are two key structures involved in these processes. The temporal lobe, which is the side of your brain, has these two structures, the hippocampus, which was mentioned earlier, and a neighboring structure called the amygdala, buried deep inside your temporal lobe. And we now know that the hippocampus is crucial for forming memories of facts and events, names, phone numbers, and daily events. And the amygdala is crucial for forming and storing memories of emotional events, such as fear memories. So the question that we've been trying to address in my lab in Bangalore is what is different between these two structures such that they can subserve such dramatically contrasting kinds of memories. To address this question, we took a strategy which involves using behavioral stress as opposed to what you'll hear about in the next talk, which is more at the cellular level. And there are two reasons for using behavioral stress. The first one is that if you want to study a difference between these two structures in terms of some processing, it's a good strategy to make it as big as possible. And that's what stress does. If you subject animals or humans or primates to chronic stress, this asymmetry gets further amplified because there's a severe deficit in factual memories, depending on the hippocampus, whereas at the same time there's an amplification of emotional memories. The second reason has to do with the fact that the stress response of your body, which is triggered by the hypothalamic pituitary adrenal axis, which ends up secreting stress hormones such as cortisol in your body, that regulatory process is under the control of both of these structures, but again, in opposite fashion. The hippocampus exerts a negative feedback control, thereby trying to suppress secretion of cortisol, whereas the amygdala has a positive drive on the same axis. So, in terms of both its response to the stress and its ability to regulate stress, in both counts, the hippocampus and amygdala appear to be going in opposite directions. So this we thought was a good starting point. And the overall strategy that we use involves looking at both structures at multiple levels of neural organization, starting from behavior at one end, neurons, synapses, and down to the level of molecules and genes wherever possible. And we use behavioral stress more as a top-down perturbation to these two networks. And whenever possible, we use knockout and transgenic mice also as a bottom-up perturbation to the genetic manipulations and then see how this pans out at higher levels. So I'll give you examples of both today. We are benefited by this analysis from the fact that there's about 2 or 3 decades of data, very rich data, coming all from the hippocampus, which has been a study that has been intensely analyzed in the business of neuroscience for over the last few decades. And in the hippocampus, what people have found with stress is the following. Any kind of chronic or severe stress leads to cognitive deficits, such as memory loss. And patient studies show that there's actually a physical reduction in the volume of the hippocampus caused by degeneration in the hippocampus. Further down at the cellular level, there's shrinkage of dendrites, again, a key marker of neurodegeneration. And further down, synaptic plasticity mechanisms that allow the synaptic strength to be changed, either increased or decreased, is also impaired in such conditions. And further down at the molecular level, there are many, many molecular markers which seem to go downhill as a result of such degenerative changes. So taken together at the cellular and molecular level, these are hallmarks of neurodegeneration, and these are always correlated with significant functional loss at the behavioral level. But all of these downward arrows, which are related to the hippocampus, are unlikely to explain the fact that in stress disorders, you also have heightened emotionality, abnormally high fear and anxiety. For that, we have to turn to the other structure that I mentioned earlier, and that is the hippocampus, and that is the amygdala. And it is now very clear, even from patient studies, that many symptoms of mood and anxiety disorders apparently have a lot to do with plasticity in the amygdala. So the question then narrows down, in our case, to studying the effects of chronic stress on the amygdala. And the amygdala is a difficult structure to deal with as compared to the hippocampus, because the hippocampus shown here in a coronal section of the rat brain. So this is the cortex, this is the midline, and this is where the hippocampus is. And even to an untrained eye, you'll see this nice layering here, which suggests to you that there's a single homogeneous population of neurons lined up in a neat anatomy. And that makes life a lot easier in terms of cellular and circuit analysis. In contrast, the amygdala is a collection of blobs of cells in multiple nuclei with no apparent order to them. And that is what has impeded detailed analysis in the amygdala as compared to the hippocampus. In today's talk, I'm going to focus largely on one nucleus of the amygdala, which is the input interface of the amygdala, called the lateral and the basolateral amygdala. And I'll give you data from multiple levels, starting from neurons and starting from behavior, then neurons, synapses, and molecules. So the first finding that we had, which is basically confirmation of previous results, had to do with the fact that if you take rats, adult rats, and subject them to chronic stress, in this case chronic immobilization stress, meaning we immobilize the rats for 2 hours per day for 10 consecutive days, and then we do our analysis 24 hours after the termination of the stress on the 11th day. The first analysis is a behavioral one, where we put these rats, the stressed and the control rats, in what is called an elevated plus maze. The plus maze has two arms. One is completely open and well lit. The other one is dark and covered up. Now, these rats obviously like to hide in dark places, and the more anxious they are, the more time they would spend in these closed arms as opposed to the open arms. So the less anxious they are, the more time spending exploring the open arm. So with this analysis, you can get a direct measure of how anxious these animals are. And what we find is that after 10 days of stress, there's a significant reduction in both the time spent in the open arm and the number of entries into the open arm, suggesting they're spending a lot less time exploring the open arm as a result of chronic stress, suggesting that they're more anxious. The next analysis is down to the level of single cells, and here's a quick primer on neurons and their architecture. Neurons have cell bodies and dendrites, which are branches, like tree branches, extending out to get information from other neurons. And these are called dendrites. And these dendrites then receive, in this case, these dendrites then receive inputs from other neurons via a structure called the axon, which conveys the action potential, which eventually leads to synaptic transmission from neuron A to neuron B. So we first analyzed the dendritic architecture of these neurons, and what we found was that pyramidal cells, shown here in the amygdala, actually grow bigger. Their dendrites grow bigger. They have more branches and more length of dendrite following 10 days of chronic stress, which is quantified here as dendritic length in microns, in steps of 20 microns radiating from the cell body outwards in each direction. And this clearly shows the upward shift of these gray bars shows that chronic stress then leads to a significant increase in dendritic arborization in these amygdala neurons. Why is this surprising? Because there was 20 years of data before us which showed that the same kind of stress actually causes shrinkage of neurons in the hippocampus. And we confirmed this finding as well. In the same brains, when we moved to the hippocampus and looked at the same class of pyramidal cells, we actually found a significant shrinkage in the dendrites. So the same top-down perturbation affected the same class of neurons in opposite directions depending on their location, whether they were in the hippocampus versus the amygdala. So this then gave us the first sort of putative correlation between stress-induced increase in dendritic arborization with increased anxiety. The next stage of analysis requires a finer resolution. So now we go and look at the dendrites in a little more detail. As I told you, these dendrites receive input from other neurons via junctions, specialized junctions called the synapse. And in the brain, these synapses, the contact point between two neurons, is actually formed on specialized protrusions, shown here, these things, which are protrusions coming directly out of the dendrite, and these are called dendritic spines, like they're little thorns which protrude out, and that's where the synaptic contact is made. So one simple way of getting an idea of what is happening to the synapse is to simply count the number of spines on these dendrites and get an indirect measure of synaptic connectivity. So the next question was, what happens to the spines on these dendrites in the amygdala? So here's a neuron in the amygdala. Here are the spines. And what we find immediately is, compared to the control unstressed neurons, there's a significant upward shift in the number of spines per unit length of dendrite in these neurons. There's a significant upward shift in dendritic spine density. So chronic stress then leads to the formation of new spines in the amygdala. So taken together then, what you have is, you have longer dendrites and you have more synapses per dendrite. So the overall increase in synaptic connectivity, the structural basis for synaptic connectivity in these amygdala cells, is greatly enhanced following chronic stress. And this is in sharp contrast to what people have observed in the hippocampus, where all of these measures go downhill in the hippocampus. So what about, we have nailed this part now, so what about molecular mechanisms? I'll give you one example of a potential molecular player in this whole business, and that has to do with the molecule called brain-derived neurotrophic factor. As the name suggests, this molecule is involved in neurotrophism, meaning helping cells to grow, neurons to grow. So when the brain develops, BDNF is a key molecule which sustains and facilitates growth of dendrites and formation of new synapses. in the adult animal as well, it is a key player in changing synaptic strength. The focus on BDNF came from work done by Eric Nestler and Ron Duman when they were at Yale, where they suggested that when you see neuropathology related to stress disorders, what is happening in the hippocampus is the following. You see a reduction in dendrites and synaptic connectivity only because you have a shortfall, a reduction in BDNF in these neurons. So they proposed the so-called neurotrophic hypothesis of depression, a key stress disorder, where they suggest that a deficiency in neurotrophic support may contribute to the hippocampal neuropathology during stress and depression. And they have much data to back this up. Some of these findings are as follows. In addition to these factors where people have shown that there's high level of cortisol, stress hormones in depression patients, some of the antidepressant treatments also can reverse these things. The basic finding is as follows. In addition to stress causing shrinkage of dendrites in the hippocampus, it also lowers BDNF. And if you now take an antidepressant, such as an SSRI like Prozac, you can reverse not only this dendritic shrinkage in the hippocampus, you can also prevent the reduction of BDNF. And these and other findings have led to this hypothesis. So we wondered what this would do for the amygdala, because this hypothesis was entirely anchored in the hippocampus. So we took advantage of a mouse that was generated in Susumu Tonegawa's lab at MIT, with whom we collaborate. And he had a transgenic mice where, through genetic means, they had been able to increase or overexpress BDNF in the brain. So these mice are basically loaded, their brains are loaded with more BDNF than normal. And then we went back, took these mice and asked the same questions at these two levels. So first thing that struck us was the following. When we do a test of anxiety, similar to the one I already told you about, now this one doesn't have a plus maze, but it has simply an open field, and the center is well lit, and the perimeter is dark. So the more anxious the animal, the more time it would spend in the margin, and vice versa. If it's less anxious, it's going to go out in the center. So if you now look at the percentage time spent in the margin, you can see that the transgenic mice, which have more BDNF, so increasing BDNF leads to more time in the margins, meaning they're more anxious. Okay. Now, mind you, this is just overexpression of BDNF. These animals have not been subjected to stress. So just the genetic manipulation of upregulating BDNF causes more anxiety. If you take control animals, the non-transgenic animals, and subject them to the same 10 days of chronic stress, you get the same increase in anxiety, which is comparable to what you got by increasing BDNF. So either stress or upregulation of BDNF seems to be giving the same level of anxiety. If you now take the transgenic mice with more BDNF and subject them to further stress, you don't seem to get much more. So BDNF overexpression seems to occlude further increase in anxiety after stress. That's at the behavioral level. If you go to the cellular level and count spines again, chronic stress in the control animals, as you would expect, increases spine density in the amygdala, as we had shown in rats. But if you take the BDNF transgenic mice and subject them to stress, you don't get any further increase in spine density, which again then suggests that genetic overexpression of BDNF seems to occlude spine formation that you would normally get with stress. So this, taken together, both of these suggest that there's a convergence. Whatever stress does, BDNF seems to do the same thing. So they must be using the same pathways. If you take all the four groups of animals and do a simple take a look at the correlations, so you now plot percentage of time spent in the center and the number of spines on the dendrites. The higher the number of spines, the lower the time spent in the center, meaning more anxious they are. So there's, again, a very strong correlation for both kinds of dendrites between spine formation and enhanced anxiety. So what we get now, what I had shown you earlier with behavioral stress, we get the same thing with the molecular manipulation, that if you somehow manage to form more spines, you get higher anxiety. But that's counterintuitive, contradictory to what I just told you. The neurotrophic hypothesis suggests that if you have more BDNF in the brain, that should be good for your brain. It should help you prevent stress-induced damage. But I remind you that that hypothesis, again, was based on the hippocampus and not the amygdala. So we had the burden to actually go and look at the hippocampus as well, because there would be a lot of skeptics in the field who wouldn't believe this result. So I'll give you a little bit of data on the hippocampal side by just looking at dendritic damage again in these transgenic mice. We took the control mice, the non-transgenic mice, and we showed that 10 days of chronic stress indeed lowers dendritic branching and dendritic length, as you would expect. So there is dendritic atrophy in the control mice, but in the transgenic mice, that atrophy in the hippocampus is completely prevented. So that key part of the neurotrophic hypothesis, which suggests that if you have more BDNF, you should prevent neurodegeneration at the cellular level, still holds in the hippocampus. And we have then gone on to look at behavioral analysis of depression in these animals, which I won't talk about, to show that indeed depressive symptoms in these animals in the BDNF transgenic mice are also lower. So the neurotrophic hypothesis holds just fine on the hippocampal side. But at the same time, when we have reduced depressive symptoms at the behavioral level and reduced degeneration, the amygdala seems to be going in the opposite direction because there's more anxiety. So by manipulating the same molecule, we seem to be getting contrasting phenotypes depending on which structure we're looking at. So, and this has interesting consequences because there's a lot of controversy now in the field, which you may have heard about especially teens and younger folks taking SSRIs, such as Prozac, which leads to suicidal tendencies in younger folks, heightened anxiety during the early stages of treatment. And we believe that, and this has led to warning labels in Prozac, for example, in the US and the UK. And we believe that some of this may have to be with the fact that the changes in the amygdala have not been looked at more carefully. And it could actually have reduced depressive symptoms and heightened anxiety coexist, as counterintuitive as it sounds. So, coming back to the key question then, I left you off with the fact that both with molecular and behavioral manipulations, we get this strong correlation between formation of new synapses and enhanced anxiety. So what's so special about these new synapses that you're forming in the amygdala? To answer this question, we then go and look at synaptic transmission specifically. I told you that these spines house the synapses, and the synaptic transmission involves an electrical pulse called the action potential arising at the terminal of an axon, triggering release of transmitters, which in this case is glutamate, and this glutamate diffuses across the synaptic cleft and goes and binds to specialized receptors, which open up, pass ions, and give rise to a signal, electrical signal in the next cell, which is called the synaptic potential or the synaptic current. That's the essence of synaptic transmission. So to look at synaptic transmission in these neurons, we resort to a different technique altogether. We cut slices of the amygdala. These are brain slices from rats, which are 400 microns thick, and we keep them under microscopic infrared microscopy so that we can look at this slice at high resolution and look at the lateral amygdala and pick out individual neurons, which in this case is a pyramidal cell. You can see the triangular shape of the cell body, a dendrite protruding from here, and this little shadow over here is what is called the whole cell patch clamp electrode, which has about a tip size of about a micron. You can go and patch onto this cell and then excite it and evoke action potentials, which tells you sort of the basic properties of the neuron and confirms that these are indeed the neurons that we want to look at. And then you can go and look at synaptic transmission, both excitatory and inhibitory, by stimulating identified fibers coming into these cells. So glutamate in this case and in the hippocampus has the option of binding to two broad classes of inotropic receptors. They're called NMDA and AMPA receptors, named after exogenous chemicals which are used to characterize them. Without going into details of the electrophysiology, all I want you to know is that these two receptors serve very different functions in terms of synaptic transmission. The AMPA receptor, which is active and visible at negative holding potential, which is close to the resting potential of the cell, which is about, say, minus 60 or minus 70 millivolts, is responsible for basal transmission, sort of housekeeping transmission of synaptic transmission in these neurons. The NMDA receptor is sort of a strange beast because at these negative potentials, it's not visible. It doesn't open up. It only opens up when the cell is sufficiently depolarized or excited. So if you take the same cell to plus 40 millivolts, for example, then you can now look at the NMDA current opening up. These are called excitatory postsynaptic currents in the order of hundreds of picoamperes. So through a clever manipulation of voltage clamping and pharmacology, which I won't go into, you can essentially look at either the AMPA current or the NMDA current in the same set of synapses. And if you do that, you can then now go and measure the peak value of these currents, this one, the NMDA peak EPSC and the AMPA peak EPSC, and get a ratio. So that ratio turns out to be about a little under 0.4 in the case of neurons taken from unstressed control animals. If you now repeat the same experiment and take slices from stressed brains, what you see immediately is that the NMDA current more than doubles in size as a result of 10 days of chronic stress. So per AMPA current, because this is a ratio, double ratio, per AMPA current is a lot more, almost double the amount of NMDA current. Why is that exciting? For the following reason. For the last 20 years, neuroscience has focused extensively on this receptor because this is the key receptor for forming new memories in the brain. And most of these findings have come from the hippocampus, where it's been shown that mice or rats, wherever you block NMDA receptor function, you'll block the ability of these synapses to change as a result of experience, and thereby their ability to store new memories. So if you now suddenly have a bulk load of new NMDA receptors on these newly formed synapses, its implications are pretty clear. The implications are such that you should now expect that if you have more NMDA receptors, which are the substrate for forming new memories, you should, in principle, have more memory, or the capacity to form stronger memories. And I remind you that the amygdala is involved in forming emotional memories, such as fear memories. Then one would predict that as a result of this stress, you should indeed get stronger fear memories. Is this a pointer as well? At the top. Great. So we now go and test this prediction that since you have more NMDA receptors, we want to find out what happens to behavior. To do that, we make use of, again, a very robust learning paradigm, which most of you are familiar with. It plays on the theme of Pavlovian classical conditioning. You remember Pavlov's dog, where if you ring a bell and follow it by food, a few of these bell-food pairings, and then you come back and just ring the bell, the dog starts salivating. It associates the two. So this is the basic form of sort of learning and memory, which has been around in the field for a long time. In rats, what we do is ring the bell, but instead of giving it food, we give it an electrical shock to the foot. And just because that's a far more powerful memory, it's an aversive memory. So you first put in the rat in a box and play the tone alone. That's harmless. The animal doesn't fear that. Then you go and pair a few of those tones, as few as one will do. But we do 5 pairings, a tone shock pairing, come back a day later and give the tone alone, and the animal freezes. And the freezing response is a direct measure of its fear memory. The more fearful it is, the more freezing. So what we now do is 10 days of chronic stress, and then the next day we condition the animal for pairing 5 tones to foot shock, 5 of these paired with this. And that's the data shown here. This is percentage of freezing, more fear, 5 trials of pairings of tone and shock. You can see that both stressed and unstressed animals end up learning this memory fairly well. They acquire this memory fairly well. They reach roughly the same level of freezing after the 5 pairing protocol. A day later, we test them in a different chamber from what they had experienced the previous day. The reason for this is that we don't want them to freeze to the context. You've come into a room and had a bad experience. If you come back to that room, you're going to show a fear response anyway. So we want to take that part out because that sort of spatial memory, in case of rats, depends on the hippocampus. We want to only understand what happens to the amygdala. So to do that, we give them an entirely different context. In fact, we give them their home cages to increase their comfort level, and then just play the tone and see how they remember the tone. So now they're being presented the next day with 3 of the same tones that were used in the previous day for training. And what we'll see here is 1 unstressed and 1 previously stressed animal. When the tone comes on, the light will come on here. That will tell you when the CS, the conditioned stimulus, is on. Clearly this guy doesn't care much, whereas this guy is frozen in fear, so to say. So data taken from such experiments then show us here on the next day when we're testing with the tone alone that the same caring protocol, the same emotional experience that leads to only moderate levels of fear in the unstressed animal leads to a very robust, very heightened level of fear in the previously stressed animals. So they're really scared when they shouldn't be, which is the essence of anxiety disorders. You are constantly fearful without any clear stimulus and clear reason. So this is one aspect of anxiety disorders which is indeed captured in these animal models. So what I've shown you today is that the same top-down perturbation can lead to a whole bunch of neurodegenerative changes in the hippocampus, all these downward arrows from the behavior level down to the molecules. At the same time, in the amygdala, we get just the opposite. We get all of these going upwards. And this then shows you that, as the first speaker mentioned, that where the plasticity happens, where the change happens seems to matter. So neighborhood matters, location, location, location. I guess in Southern California, those who live in Southern California don't have to be told this. Their real estate agents must have told them that already. So that's what we find in neuroscience as well, is that the same set of players from the molecules to the cells seem to be in play here, but with very dramatically different consequences. And I'd like to point out then that if we had these changes that we see in the amygdala on the left-hand side, if we had the same changes in the hippocampus, those would be termed as good plasticity, because it's been shown that if you give, instead of a stressful environment, if you give an enriched environment, lots of toys, lots of social interaction, a happy environment, indeed, the same changes happen in the hippocampus. All these upward arrows show up. And if you had these changes in the hippocampus, you would actually be able to counteract some of the neurodegenerative changes that are induced by detrimental conditions. And indeed, some of the Alzheimer's models have been has been used to show that you can reverse some of the Alzheimer's pathology, for example, by putting animals in an enriched environment. So these changes in the hippocampus would definitely be considered as good plasticity. But when they happen in a bad neighborhood, such as the amygdala, they have detrimental consequences at the functional level and give rise to symptoms which are common in stress and anxiety disorders. And that's the same thing we had also arrived at by our molecular perturbation using single gene manipulation of the BDNF molecule in both these areas. So with that, I'll end and thank the folks who did all the hard work, past members of the lab who started some of this work, and current members, graduate students who are now doing much of the behavioral work. And we're always thankful to our collaborators at MIT and other collaborators that I haven't mentioned today and our funding sources. Thank you very much.